discussion, you can put no group in front of your name today. Also, I must apologize. I have very severe kafunjo. So maybe I don't look so great. I would like to turn my camera off, but I cannot. So if I look terrible today, it's because of that. If I have to take some breaks during the class, please forgive me. And if a few of you would volunteer to put your cameras on during today's class, that would be very nice to be able to have an audience to speak to. Just a few of you is fine, a few brave souls. Thank you very much. Okay, a couple more people are joining.
Recording in progress.
Okay, so welcome to the second day of the course for scientific presentations. Today we are going to focus on really using context, providing context to the audience at the correct timing and in the correct way that they can deepen their comprehension of your research story. I've included some definitions of context here on the screen. I think you know context. Even in Japanese, we use the word konteksto because there's not a really good Japanese word for it. You can see here the Cambridge Dictionary has defined it as the situation within which something exists or happens, and that can help explain it. And this last part is really important, right, for context, that in order to deeply understand a concept, we need to understand the situation in which it is happening. And Oxford it's a very similar definition, but slightly different words. The circumstances that form the setting for an event, statement, or idea, and in terms of which it can be fully understood. So context is necessary in order to achieve full comprehension, for us to know how to use the information we are receiving. For example, on your screen right now, you probably see this strange yellow plastic piece here, and you're probably wondering, what is this? Why am I looking at this? And of course, without any context, there is absolutely no way for you to know what this is, why you are looking at it, or what to do with it. This is actually a piece of a children's toy from when I was a child. Maybe now this is no longer given to children, but we had this ball like this with holes in it and all these pieces. If I were to give you this first and give you a moment to think about this, and then I give you this piece here, your reaction to this piece would be very, very different. The moment you see it, you would instantly know how it fits into this larger picture here. You would know what to do with it immediately. And that's the same thing with information or with ideas or data that we provide in our presentations. And unfortunately, this is something that many presenters do not consider when they are designing and writing the script for their presentations. So these are some questions that are very common, but these are absolutely bad situations for a presentation. We do not want our audience to be thinking, why am I listening to this? How do I process or use this information? Which details am I supposed to focus on? Or how is this related to the goal of this presentation, to the key question? These things should always be clear at all times. So please take a moment and put this in your minds, that this is a major goal of creating your scientific presentation, to design it in a way that the audience, as they receive any explanation or data, they always understand why they are listening to it at that moment, how they are meant to process and use that information which details they should focus their attention on, and how is it connected to answering our key question. So yesterday we talked about the principal goal of scientific presentation is to get meaningful feedback from our audience. And in order to get meaningful feedback, we need the audience to deeply comprehend many key concepts and parts of the scientific presentation. Specifically, what is your research goal? How do you feel about that question? Why is it important or significant? The logic, the motivation for your experimental strategy, the significance of all your results and how they are connected to the goal, and the logic and the importance, the significance of your conclusions. So all of this we need to get from our mind into our audience's mind. The audience, as I said yesterday, as an audience of scientific peers, will be using their own scientific background and experience and education to imagine that they are participating in the research story, to make predictions and participate in an interactive kind of way, doing critical listening. But in order for them to be able to do that, we must provide them with the correct context first. So there's a lot of barriers, obstacles to deep comprehension. When things in our presentation are unexpected, feel unconnected, unfamiliar, unclear, too complicated, maybe too fast, or simply because our audience is human, they might be daydreaming, thinking about something else. This is very common. Almost all of these, providing the correct context at the right time, will be able to overcome almost all of these difficulties. So let's immediately go into an example. I would like you to think about the explanation of a slide, two different versions, and focus on what is different about these two versions. I gave you all pre-lecture slides, so I think you already kind of know the answer to these, and that's fine. So for our first one, we have some background information. It is not the first slide in the presentation, so we imagine that the audience already knows that the presenter is studying various characteristics of a specific type of cancer cell and the ability of those cancer cells to invade different organs in the body. Before this slide, he or she explained that the cancer cells grow rapidly in the lungs and that they easily spread to the bones, the pancreas, and the intestines. So I'm going to present this slide, two different versions, and I think the big difference should be obvious to you. But as you listen to them, think about which version is easier to predict what is going to come next, and which version requires the audience to use more of their mental energy to focus on the details and try to comprehend. So here is version 1. We next examined the spread of cancer to the liver. In this case, the cancer cells, colored in red, spread from the lungs and reached the upper border of the liver, but did not penetrate it. In a separate case, cancer cells approached the liver from behind, but also stopped before invading it. In yet another case, the bottom part of the liver was also inaccessible to the cancer cells. This seems to indicate that these cancer cells cannot spread to any part of the liver. We don't know why the liver is protected from invasion, but this information would probably provide a clue for the development of effective treatments. Okay, that's version 1. But let's move on to version 2. It's exactly the same slide, so I'll just go back to the animation. Okay. An important finding is that these cancer cells cannot spread to the liver. This is true no matter which part of the liver is approached by the cells. In this case, the cancer cells, colored in red, spread from the lungs and reached the upper border of the liver, but did not penetrate it. In a separate case, cancer cells approached the liver from behind, but stopped before invading it. And in yet another case, the bottom part of the liver was also inaccessible to the cancer cells. We don't know why the liver is specifically protected from invasion, but this information would probably provide a clue for the development of effective treatments. Okay. And I'll show you one more example from a different field, although I think this example is probably pretty simple. In this second example, the researcher is studying the molecular clouds in space where stars are born. So they are born from dense cores at the center of molecular clouds. And the cores are often round, but sometimes they are elongated. The presenter is looking into a potential relationship between the shape of the cores and the number and orientation of the stars that are forming inside of them. The shape of the core is determined by measuring flux values and drawing contour lines of equal flux values. Here's version 1. This is an image of a core with an embedded binary system. The two white spots are embedded stars, and the blue line is the 50% flux contour found by connecting points with half the flux value of the peak flux for that core. About half of the binary systems we surveyed were associated with elongated cores. At first, we thought that the embedded binaries might affect the density distribution inside the core, thereby causing the elongation. However, we found that many starless cores were similarly elongated. Based on this observation, we speculate that elongated cores that host binary systems were already elongated prior to star formation. In order to clarify this and identify potential mechanisms for the elongation, we plan to do a systematic survey of starless cores. Let's look at version 2. Our results suggest that cores may be elongated at early stages before any potential influence from embedded primary systems. We determined the shape of cores based on the 50% flux contour calculated from the peak flux value and shown here as a blue line. We found that about half of the cores containing binary systems, like these with embedded stars shown as white spots, were elongated. We thought this might indicate that embedded binaries affect the density distribution inside the core, causing the elongation. However, we found that many cores without any stars were similarly elongated. To determine if cores are indeed elongated prior to star formation and identify potential mechanisms, we plan to do a systematic survey of starless cores. Okay, so in the chat, I would like you to, hopefully I think everybody knows the answer, but in the chat, just type very quickly, what is the main difference between version 1 and version 2 in both of these examples? What is the primary difference between the speech in version 1 and version 2 for both of these? Okay, not an overwhelming number of answers, but the answers I'm getting are all saying the same thing, aren't they? Okay, they say conclusion, or hypothesis first. It depends on where we are in our story. In this case, it's generally the conclusion or the hypothesis. But for the sake of this slide, if we consider one slide to be one story with a message, something that we want the audience to remember, to learn from this slide, to take away from this slide, we can say that message is the conclusion of the slide, right? Not necessarily a conclusion of the research experimental data, but the conclusion of this slide comes first. Okay, so yeah, this is a really key idea for comprehensive presentation, is to put the conclusion first. And this is something that a lot of presenters do not do, and especially Japanese presenters often avoid. Well, not just Japanese, but East Asian presenters in general. So Japanese students often start with version 1, where we begin immediately with specific minor details, especially telling the audience what they see in the slide or what they see in the data. And that means that as we are listening to the explanation, we don't have any context. We're immediately beginning with fine details. And the audience doesn't know yet how they are supposed to use or interpret that information. It means that the listener must, as we are listening, we must memorize all of the information first. And then at the end of the slide, when the presenter finally tells us the message, we now have to go back and remember the information and decide, do I agree that that information supported this message? This is a very difficult way to listen. It is not impossible because human brains are pretty smart, but it is going to use up your audience's energy very quickly. And after a few slides like this, people will become tired and stop focusing on your presentation. They will switch into a passive listening mode where they just kind of let the information flow in front of them and don't really try to listen. Another key problem with this is that as we are listening, it's very difficult to distinguish between key data or key information and supporting information. All stories, well, I mentioned yesterday, we need to remove as much as possible, right? We need to be strict with ourselves and remove things that are not necessary. But we will still have key information and ideas that we need the audience to focus their attention on, to think about, to remember, maybe to take notes about. And there will also be some supporting information, which is necessary for comprehension, but the audience doesn't need to use their mental energy to think about or remember. But if we don't know yet the conclusion from the beginning of the slide, if we don't know the message yet, how we are meant to use the data or what we are meant to conclude from this information, we cannot distinguish between the key information and the supporting information. So we're going to be concentrating very hard on everything, using our memory, recalling it at the end of each slide quickly to try to establish a connection between the information and the conclusion before the slide changes. This is very tiring. The second version, however, for both of these, but the conclusion comes first. The conclusion is unmistakable. It means that as we listen to the data, if we hear the answer first, then as we listen to the data, we can interpret it in real time. We can look at the data and decide, do I agree that this supports the main message, that this supports the answer? Do I understand how this information supports the answer? And remember, to give critical feedback, that is extremely important to be able to do this. It also allows us to distinguish between key information and supporting information if we already know the conclusion. We can recognize this is really important, this is just supporting information. And that we're not taking a bunch of notes about information we don't need to remember later. You might also notice that I read the second versions a little bit faster. That was not because I think you're bored. It's because if you know the answer first, you will be able to comprehend the contents of that slide more quickly. And so it's a little bit, it's also going to save some time. And this is really key also for saving mental energy. And you can keep it for later in the presentation. So here's an idea here is saving time and saving mental energy for later, because almost all presentations, your key results, your most important experiments usually come at the end of the presentation. And the experiments that come at the beginning are often preparing us to listen to and comprehend the significance and importance of those important experiments at the end. 必ずしもではないですけどね. That's not always the same structure, but very often. And so we have this distinction between minor experiments and key experiments. And one real big difference is for minor experiments, maybe you don't really need the audience to listen very critically, to use their scientific knowledge and background to judge, do I agree with this? But your newest, most important experiments, you do want the audience to have an open mind and use their mental energy to deeply comprehend at least those experimental results and decide, do I agree with this? Do I have any alternative ideas, etc.? It would be nice if our audience can do that for every experiment on our presentation, but humans are limited with our energy. So for key experiments then, we might actually choose a slightly different strategy. And the reason is if you give the conclusion 100% of the conclusion first, of course you do create a bias in the way that we view the data. And this bias could potentially prevent the audience from thinking of any alternative explanations or alternative interpretations. So for the key experiments, if we want to prevent that kind of prediction bias and allow the audience to think of more ideas, what can we do differently? Well, we still want to emphasize the importance. We want the audience to feel that this particular experiment or this portion is important. We want to make sure they still have a listening goal. They know precisely what kind of answer or what kind of information they will get by the end of that slide or by the end of that explanation. And of course, we want to do it in a way that we can still invite open interpretation. So we want to basically stimulate critical thinking for this portion. So I'm going to show you a third example that's slightly different, and I would like to see if you can pinpoint what is the strategy in the second version for this example. Okay, it's slightly different. It's not the full conclusion. So a background again for this. This is an archaeology example, Kobogaku. The presenter found some Edo period maps, and they believe that these maps correspond to a recently discovered archaeological site. So he hopes to find evidence. The sentence here is incorrect. He hopes to find evidence from the site that would correspond to the dates on the map, and therefore support the idea that the maps are for this location. That's what it should say here. So the dates on the maps of the 16th century. So basically he is hoping to find evidence from the site, from the location that matches those dates. Okay, here's version 1. In order to provide archaeological evidence to confirm the dates found on Edo period maps for Site X, Area A was excavated. Several coins were unearthed, but the characters written on the coins could not be easily read. They were examined with X-ray CT equipment and revealed to have been produced and circulated since the late 15th century. Although this did lend strong support for the Edo period maps that dated the site to the 16th century, this also provides compelling evidence for economic activity in Site X at these early dates. Such early economic activity had not previously been reported for this region. Okay, version 2. We made an important finding at Site X that changes our historical understanding of the region. Initially, we only hoped to find archaeological evidence to confirm the dates found on Edo period maps for Site X. But when we excavated Area A, we unexpectedly discovered these coins. As you can see, the characters on the coins cannot be read, but X-ray CT scans determined that they had been produced and circulated since the late 15th century. Now, although this did lend strong support for the Edo period maps dating the site to the 16th century, this also provides compelling evidence for the first time that economic activity in this region began much earlier than previously thought. Okay, so what is the big difference here? This one is a little bit more difficult, but in the chat, I'd like to see if any of you can pinpoint what is the strategy here used to introduce the slide. It's not the full conclusion, right? In the chat, if anybody has any ideas. So far, I have one answer. State the importance of the finding, and then go on to reveal the data. And this is absolutely a part of it, right? We state how important the finding is, but we don't tell exactly what the finding is, right? We say that we found something. We say that it is important. And there's something else as well. We also give a specific hint about what kind of knowledge we gain from it. We don't specifically say what that is, but we give some pretty big hints. So in the speech, I said, we found something important that changes our historical understanding of the region. So I tell you that we are going to change history. Something that we have believed about this region historically, we're going to change that. And that's pretty big. It emphasizes certainly the importance of that, but also tells us something specific and can encourage the audience to look and think about, what do we know about the history of the region? And as they see the data appear, for example, when they see these coins appear, they can ask themselves, what does this change about history? Why do these coins change history? And encourage the audience to think about that and use their own understanding of the history of this area to see if they can come up with what is new or interesting about this. And then there's also another hint on the slide. In the title of this slide, it says clues for economic history found at site X. So put together, we know it's not just history, but history of the economy of this region. So I don't tell you the specific thing, which is the main message is that we believe that the use of money, currency, began earlier in this region than was previously thought. But I tell you that we're going to find something that changes our understanding of economic history. And the second sentence in the speech also, I said, initially, we only hoped to find archaeological evidence to confirm the dates on the maps. This sentence, what does this tell us? Initially, we only hoped. That tells you that although we are going to tell you this answer as well, this is supporting information. This is less important information. So in all three of these strategies, I think you can tell that not only do I give the conclusion first, but especially for the cancer examples and this example, I also give the audience a hint of what kind of data we are going to see and prepare them to listen to that. For example, in the cancer example, I told you first, it cannot invade the liver. But then I also added, this is true no matter which direction it approaches the liver from. And that what kind of data did I show you? I showed you the cancer cells approaching from the top, from the back, and from the bottom. So I gave you a hint about what kind of data you are going to see. And this is a really important concept as well. It will make it much easier for the audience to listen to the data and to be able to apply it to thinking about the conclusions themselves. So this second strategy, it's a partial conclusion, right? I'm giving a part of the conclusion or a very specific information about the conclusion without revealing all of it. Once again, the first example, there's a lot of details without any context. The listener will probably mistake the main message. They will probably believe that the main message is to confirm the dates on the maps. But in the end, that is not the main message. And another thing is the way that this slide is explained focuses very much on what was done. These were unearthed. The characters could not be read. X-ray CT scans were used. It focuses very much on these items. They take a very primary position in this explanation. Whereas the second version is focused more on what we learned from those procedures. So the important finding, change history, is given part of the conclusion. The audience knows they can anticipate something. And as I said, the explanation focuses more on what we learned. So X-ray CT scans is included, but I say from X-ray TC scans, we determined that. So the sentence puts the emphasis on what we learned from the scans, not on the scans. And this is another thing that many Japanese students in their presentations, they focus the grammar too much on the experiment or the procedures and not what we learned from them. Another minor point is the second version uses you and we. It uses more conversational style of speaking, which makes it a lot easier to listen to. But we'll think about that later. So basic rules for this. Any new slide, any graph, any new data, anything new that you present to the scientific audience in a presentation, the audience wants to know, what are we going to learn from this? We need to know what is the purpose of this item first. We need to be able to know what elements inside of that to focus our attention on and how to interpret it as we listen to it. Not store it like a computer and access it later to interpret it, but interpret it in real time. And this is done by the topic sentence. So I included this in the pre-lecture information because I know a lot of students who are educated in Asia do not learn about topic sentence for writing, but it's very, very important for scientific writing. The topic sentence tells us our listening goals. It provides the context for all of the contents in that. It increases comprehension and improves feedback. So the topic sentence is the first thing you say. And it says topic, but it's really more of conclusion sentence. The topic sentence is going to give the answer first. Remember, feedback is what we're doing. So I also included in the slides this bit from Wikipedia, and just the important part here, right? The topic sentence is a really key part of writing, academic writing. It's usually the first sentence in a paragraph. It summarizes the main idea of that paragraph. And so for a presentation, each slide is like a paragraph. And we start by summarizing, giving the answer for that slide first. That first sentence should also reveal its relation to the overall thesis. In other words, its connection to the key question and the conclusions. And ideally, it will provide cohesion, means stick together. In other words, show how that slide fits into the story, its role in the story, and create a smooth flowing storyline. So all of this is done by the topic sentence. When we design a presentation, remember, our goal is maximum comprehension. It is not literature. So I know in East Asia, in school, when we learn to write, we learn Shotenketsu. And Shotenketsu is very wonderful for creative writing. However, academic writing is not creative writing. At creative writing, we want to use suspense and surprise and make people think more deeply about an idea. We give them the topic first, and we develop it in parallel to what we're really talking about. And then we reveal that, in fact, this is the true meaning behind all of this, and makes the reader go, oh, wow, that's so interesting. And then we tie it together. That's Shotenketsu. And it's great for novels, for literature, for movies, for romance, things like this. But a scientific presentation, we want the audience to catch what are our goals. We want them to be able to appreciate our scientific genius of creating a strategy to answer our goals, to answer our question, and to comprehend what evidence we have and the logic to get our answers. Romance, mystery, suspense, we don't need these in scientific presentation. One exception is the introduction. So I think you can use Shotenketsu to introduce your key question at the beginning of the presentation, to make a stronger impact about it and help the audience understand the significance of it. In a way, the perspective frame is very, very often done Shotenketsu, very successful. But for the rest of it, please forget about this writing style that you learned in school. So my Japanese students often feel a very strong resistance to putting the conclusion first, because they were taught you don't do this. But for scientific presentation, for scientific writing, it's the opposite. Our goal is not literature, it's to maximize comprehension. So a lot of students then say, okay, topic sentence. Next I analyze my next experiment was 90. These are the results. These are not topic sentences. The topic sentence is supposed to give the conclusion, the main message. The main message of these sentences then is that you did an analysis. So when the audience goes home tonight and remembers your presentation, do you want them to remember that you did an analysis or that you have results? Of course not. That's not what you want them to remember. You want them to remember, of course, what you learned from those. So these are really common. We hear these all the time. And I think students hear their senpai, or unfortunately, even their professors, give presentations like this and they imitate it. So we do commonly hear this, but it's not effective. It does not prepare the listener to deeply comprehend the contents of the slide. And very often, this pattern focuses too much on the procedure of the experiment and not enough on what we learn from it. So in order to provide context, you want to think, what is the real main message of this? And this is something I find students don't think about. When I critique my students' presentations, sometimes a slide it looks like a list of information, and I cannot feel what is the message of this slide. What am I supposed to catch and remember and use from this slide? And I'll ask the student, what is the main message that you want your audience to get from this slide? And they often say, I don't know. I didn't think about that. I needed to use this data, so I organized it in this slide. So it's maybe a quick way to make your presentation, but it's not a good way for the audience to understand. So you really need to plan your presentation knowing that each slide has some message and designing the slide around that message. Okay, and that message is, what do we learn? What are we supposed to remember from this slide? And put that first. Okay, it's pretty simple. Just put it first. So in the pre-lecture slides, I gave you an example of giving directions to, I don't remember. It's been so long since I've been to Tsukuba. Some train station, a JR train station. And I gave you the first example, just directions. Exit here, turn this way, go this many meters, turn here. And I think you can agree that those kinds of directions are simple, very, very clear, very precise, and accurate. But if someone gives us directions like this, we will never be able to remember and use those directions. We cannot process it. Our brain is not possible to process it like that. We need to know the goal first. And as we listen, we also need some guidance. We need some connections to knowledge that we already have. And so we need context throughout the explanation to help us understand, how does this information fit into the bigger picture? How is it related with something I already know? And in that way, our brain can process the information as we listen to it. And we can kind of visualize the story, process it, internalize it, digest it, and remember. Okay, so I'm not going to go repeat these instructions again, but you can read the directions again. And I think you'll agree that if you were to give directions, michian, to anybody, you would always use the second pattern. Nobody is going to give directions like the first pattern. So in society, in daily life, you know that this is true, that you cannot just suddenly give a complicated explanation without first stating the conclusion. Okay. So the topic sentence, as I said, is really, really important. It's a must. It is essential. But it's not enough just to give the topic sentence and then do like a detailed list of minor procedures and things. So students often, especially for example, when explaining a graph, they will always start with the x-axis is this, the y-axis is this, the blue line means this, the red line means this, the intervals are measured at 1 hour. I used this equipment to get this. They give all of this explanation about the details of the graph and the conditions of the experiment, but The audience is looking at the graph and waiting for the presenter to finally tell them, what does it mean? How do I use this graph to contribute to answering our goals, our hypotheses? I think usually when students talk like this, the audience ignores them. Because, first of all, we can read, right? We can see what the x-axis is and the y-axis is. We can see what the lines are. What we cannot see is what that means. So the audience will begin to attempt to interpret the information in the graph by themselves. And very often they interpret it incorrectly. So it is really important to tell them quickly, first, what is the answer? What does it mean? What is the conclusion? And then explain in a way that helps them to connect what they visually see with the actual phenomena that happened in the experiment. So we need to not waste time talking about minor elements as full sentences. You can include the meaning of the x-axis as a small part of a sentence about something more important. So we're going to look at examples of this, but please keep that in mind, that it's not just the topic sentences is enough to fix everything. The explanation needs to be focused on the story, maintaining the context to help us interpret what we see. So we start with often purpose, the meaning. We give a hint about the kind of data we are going to see, about what to look for. We focus on the significance of the data, what it represents in the actual organism for biology or whatever in other fields. Not the labels, not just reading labels. And we need to explain the details in the context of the story. Okay, so we're going to look at a lot of examples like this later. So topic sentence, there's two types, as I mentioned. We can directly reveal the specific conclusions. This is fuel efficient, okay? It can really create easy comprehension in the audience and doesn't use too much of their energy. Or for our more important experiments, we can tell the audience some specific, intriguing information about the conclusion and let them listen for the complete answer. So it's going to emphasize the importance of provide listening notes. But whichever of these you choose, so I find the students, when they choose this second version, it ends up being too vague. Okay, and they say, oh, but this is just a partial conclusion. But it's not enough very often. So a good test for this, ask yourself this: does my topic sentence answer these three questions? What information or data will I get on this slide? What expectations should I have of this slide? What am I going to learn on this slide? These three questions should be answered by your topic sentence. So this is a really good test to know if your topic sentence is good. On this slide, I will show you the results of the experiment. It does not answer these questions. It answered about 1/2 of the first question. It does not answer the second question. It does not answer the third question. So I'm going to show you the results on this slide. That is not a topic sentence. Okay. And ideally, it should also reveal the connection to the key question and the overall story. So some sample topic sentences, I've already shown you these, but just to think about what good topic sentences might look like, they don't always have to completely say the names of organisms or whatever. We can be a little bit broader. So assuming that the slide before we clearly explained our hypothesis, predictions, and logic for what we predict, this would be a good topic sentence. As you will see, the results indicated that our suspicions or our hypothesis was correct. Although we had expected a sudden decrease, The data you will see on this slide presented us with a surprisingly different trend. This is a partial conclusion. On this slide, we'll see that the two compounds were indeed incompatible, but not for the reason that we had expected. This is also a partial conclusion. And finally, next we will see why the gene was not considered to be a candidate. This is also a partial conclusion. And one more thing that's really important with the topic sentence, and you're looking at it right now, is the slide title, the text at the top of every slide. Of course, this is the first, well, hopefully the first thing that the audience will look at, and they will use this to help them predict what they're going to learn on this slide. So the slide title works together with the topic sentence to provide context. And it has to be shorter, right? It has to fit in this little line at the top. So we usually just include a very short version or keywords only, but it should answer the question, what will I learn from this slide? So if your slide title is methodology or results or electron microscopy or the name of an organism, that's not telling the audience what they're going to learn from the slide. You should include a little more information then. Again, slide titles, there's two types. Conclusion title gives the final conclusion, or it reveals the key result. And in that case, after giving the key result first, we will usually include a second pop-up text with the conclusion written before the end of the slide. So an example of that would be like this: liver protected from invasion would be a conclusion title for this slide. And the other topic title gives specific meaningful indication of the contents that we are going to see on this slide. For example, this one with the coins, clues for economic history found at site X. This is a topic title. Okay. So another really important information we must give in the topic sentence and also continue to focus on throughout the slide is something we're calling the main character, shujinko. And this is something else that when you're planning your presentation, I want you to decide in the planning stage on this slide, not only what is the main message, but what is the key idea or topic or specific theme that you need the audience to focus their attention on in order to comprehend your message. And we're going to call that the main character of that slide, shujinko. A specific topic that you need the audience to focus on throughout that slide. It needs to come in the topic sentence, but throughout the slide, every time it appears, we want the audience to focus on attention, right? So we want to emphasize this main character every time it appears. So one way we can emphasize this is to put it in the strongest position of each sentence where it appears. So grammatically, the position in the sentence that the listener will automatically focus on. So very often it's the subject position, especially when the subject is attached to a strong verb, a key action that is happening. In that case, the subject position will be strongest, but not always. For example, sentences like these. We learned that the trial led to the discovery of the results indicated These kinds of verbs obviously create interest in the audience, and so the moktekigo, the object of these verbs, direct object of these verbs, is the position that is strongest in this kind of sentence. And the subject, we, the trial, the results, obviously these are not the important information in these sentences. So subject position is often the strongest, but not always. Okay, so we want to put the main character in places like this that are strongly emphasized naturally. So let's look at an example of this. What I want you to do is, as we listen to this next example, well, you can read with me. I'm going to show you the text. But as we listen to this, I want you to imagine that this is the introduction to a presentation. And that after this introduction, we are going to hear some very specific story. And I would like you to predict as specifically as possible, なるべく具体的に, what do you think will be the story that comes after this introduction? And after this, I'm going to put you into the small groups and have you discuss with each other, well, share with each other your ideas about what you predicted as you listened. つまり聞きながら自然と想像してみてください. Don't analyze the text after and look for clues. I just want you to listen naturally, and whatever naturally comes to your mind as what you think will be the story that comes after this. I want you to share that in your group, okay? So here is the original version. In 1928, Scottish scientist Alexander Fleming noticed that bacteria colonies cultured in a plate were destroyed by contamination with mold. His accidental finding eventually led to the historical discovery of penicillin, a group of antibiotics. Until then, there had been no effective treatment for pathogenic bacteria, whose infections were often fatal. When bacteria are treated with penicillin, the synthesis of the cell wall is inhibited and the bacteria die. Although many types of bacteria are now resistant to it, penicillin is still widely used today. Okay, so formulate that into a sentence or an idea of what story do you think is going to come after this? And I'm going to organize the breakout groups now. Okay, so I'm sorry, there were a lot of no group people, so it took a little bit of time to do that. I'm going to send you into the breakout groups now. I don't think this will take very long. I just want you to share with your idea what was your natural expectation for a specific storyline that will come after this introduction. So let's say a maximum of 5 minutes. If your group finishes earlier, you can come back to the main session.
Hello.
Hello.
How would you think about that story?
Sorry, my background is not science, so
Yeah.
I just predict like today's penicillin's useful way to use or something like that.
Okay.
How about other people?
How about Akasaki-san?
高崎さん、はい。
I think that the way to use the penicillin or another medicine like penicillin.
Yeah.
How about is it okay for you speaking, Mr. Khan? まだミュートになってますよね。大丈夫ですか?
Yes, it is okay, but this field is totally different.
.杉本さんはどうですか?
これ日本語でいいんですか? So in my opinion, I just think about that has a history, how human overcome the infection disease. Because the penicillin is the first antibiotics. I don't know. 何を話したら終わりなのかよくわかんないんですけど。
この後続くストーリーを予測すればいいんですよね。どんなことにつながるかっていうことですよね。カーンさんはいいんでしょうか。Mr. Khan, Dr. Khan, can we hear your opinion?
Yes, we can hear actually. Let's see what they are explaining. Wait a minute. This slide is not available. Can we see or not? it was okay, but the information is uncompleted.
Excuse me.
So we need to back to
Okay.
main session.
It's time to come.
Thank you.
Recording in progress.
Okay, welcome back, everybody. So this is a very simple activity. I just wanted you to share your ideas with each other and hopefully see that the different members of your group had different ideas. So I'm not sure how that worked out. Did any group have every person with the same prediction of the story that came after? Did any group have that? Yes? No, mostly it was different predictions, correct? I've done this in my class before, and here's a list of some of the predictions that we've had in my class before. I don't know why the first one is a different color. Fleming's work after discovering penicillin, antibiotics other than penicillin, antibiotic-resistant bacteria, the continued use of penicillin today, the mechanism of penicillin and other antibiotics to kill bacteria, the many members of the penicillin family of antibiotics that exist today, the mechanisms that bacteria obtain antibiotic resistance, and many more over the years. So why does everybody listen to this same introduction and have so many different predictions about what's going to come next? Well, the obvious answer is I don't provide enough focus for you to predict what I'm going to talk about next. There's too many different possibilities in this introduction. Its focus changes basically each sentence. So if you notice the colors here, I have colored the subject of the sentences in red, and the strongest, okay, it looks green too. I don't think I would have designed the slide like this. I think it changed somehow. I would never use red and green as opposing colors because so many people are colorblind to red and green. But it looks like it's green, isn't it? The green words like bacteria colonies are the strong position in the sentence when it's not the subject. And as you can see, Alexander Fleming, bacteria colonies, finding, discovery of penicillin, effective treatment, synthesis, bacteria, and penicillin, it continuously changes. There is no one focus on this. So what it means is that the audience, as they listen, they are 100% going to rely on their own experience, their own education, their own interest, and their own imagination to decide what they believe is the key information or the most important ideas in this introduction and focus on that. And subconsciously, muishiki ni, you will develop predictions based on that. And obviously, this is not a good situation for a presentation where you have a large audience and everybody is listening to you in a different way. Everybody has different expectations of what direction your talk is going to go. So we have to make a conscious effort when we are planning our presentation and when we are writing our script to focus it on the idea that we need the audience to focus on. So we need to use grammar and sentence structure and word choice to make sure that our important ideas are strongly emphasized, and that even though the audience doesn't realize it, They are being made to focus on the correct ideas. So one 中途半端 solution to this is actually the one that's in the textbook, but this is a little bit more focused, okay? Penicillin is a group of antibiotics derived from fungi. It was it was accidentally discovered in 1928 by Scottish scientist Alexander Fleming when he noticed that bacteria colonies cultured in a plate were destroyed by contamination with mold. Penicillin is historically important because it was the first drug that is effective against many pathogenic bacterial infections, which are often fatal. Penicillin kills bacteria by inhibiting synthesis of the bacterial cell wall. Although many types of bacteria are now resistant to it, penicillin is still widely used today. Okay, so I think you can see, especially by the red text, that the subject, at least the main character here, is penicillin. But remember what I said the main character needs to be: a specific topic or concept. Is penicillin a specific topic or concept? Not really. Penicillin is the name of a thing. But what about penicillin? So just this introduction here, there are still many, many different possibilities. So of our original list, maybe these here are probably not going to be predicted by the audience. But I think there are still many different possibilities that this story could go, different directions. And the reason is that penicillin as a main character, it's kind of too big. We want to make it more specific, a more specific topic, like a relationship or something. So I proposed a better main character. And here's my third version of this introduction. And I think this time, the audience will hopefully be focused on and thinking about the same topic and maybe making the same predictions. The impressive array of antibacterial mechanisms boasted by today's antibiotics began with the cell wall targeting penicillin. The bactericidal means to kill bacteria, adjective form. The bactericidal property of penicillin was discovered in 1928 when Alexander Fleming noticed mold spores killing cultured bacterial colonies. This ability to kill bacteria caused penicillin to become the very first drug effective against frequently fatal bacterial infections. Penicillin's effectiveness to target the bacterial cell wall means it is still widely used. However, many gram-positive bacteria are now resistant to it. Efforts to find alternative mechanisms to combat bacteria are still leading to the discovery of new antibiotics today. Okay, so as you can see by the colored parts here, the topic of this is the ways, the mechanisms by which antibiotics, including penicillin, kill bacteria. So this is obviously a much more specific topic. How do these drugs kill bacteria? And if you're focused on that throughout, I think your predictions for the story that comes after maybe will not be completely the same, but they should be basically in the same direction. The idea here is the audience does not need to be able to make predictions precisely about what will come next. But when the next slide or when that story does appear, it should not feel different or surprising or unnatural. It should feel natural. So if the audience is expecting a completely different story, then later when the story does develop, they will feel weird. あ、こういう話が全然なんか予想できなかった。And that's not good. Okay? あ、予想できなかったんだけど、なるほどですよね。こういうことなんだ。つって。So that's a little better, obviously. So from our original list, I think almost all of these we would not predict from that introduction. Probably either the mechanism of penicillin and other antibiotics, or we might expect it to flip around and talk about how then do the bacteria resist that mechanism and develop antibiotic resistance, I think is also still possible. Okay, much, much more narrow. This is a really important concept for successful scientific writing and also especially for scientific speech. So, let's give you an example of an explanation of a graph. And I'd like you to notice, first of all, the design. It includes like a slide title, but for the graph. Many students here will basically put blood sugar level increase over time after ingestion as the title of the graph. But that's a waste of space because the audience can already read the X and Y axis labels. Making the title the same as your X and Y axis, it's just repeating the same information twice. Remember, the graph has a very specific reason, something you want the audience to learn from it. There is a message to that graph. So try to put that message in the graph to give them a hint how to interpret it and what to learn from it. So in this case, I made the title Comparison of Blood Sugar Time Course, and then a subtitle to give the conclusion. Mannan diet is safer than Slim and Slim. So the audience knows from the beginning, the answer we're supposed to get is this one is better, it is safer than this one. And now we're going to listen to the story to see why or how we can see that. So the graph has a message. It also needs a topic sentence and a meaningful title. And the story needs to be about what's really happening, not how the lines are moving, because the audience can see that. Very often students will just tell you what these lines are doing, how they are moving. Well, the audience can see that. The audience needs to know what does that mean. So here's a sample explanation for this. I prepared two, one long and one short. The change in blood sugar level over time shown here. Sorry, I didn't include this. Well, I'll just have to remember where to. Usually in my text, I always include a note of where to click for the animation. It's a really good tip for presentation because you can't always remember when to do it. But I forgot this time, so I'll have to do it from memory. So this is a bad example, what not to do when you're prepared. Okay, starting over. So here's my graph, right? The change in blood sugar level over time shown here strongly indicates that Mannan diet, shown here by the pink line, is the better option for patients taking drug XYZ. Keep in mind that our primary safety concern for use together with XYZ is avoiding rapid and large changes in blood sugar, which can be very dangerous for the patient. Even casual observation of the Slim and Slim blood sugar time course shown in the blue line can tell you that it's not very good. The maximum acceptable increase is 15% over 10 minutes. Not only did Slim and Slim reach the maximum limit for 2 consecutive 10-minute periods here and here, the 20% increase seen from 60 to 70 minutes after ingestion exceeds this important limit. The erratic fluctuation seen in the middle portion of the plot is also cause for concern for patient health. On the other hand, the smooth and conservative increase and decrease in blood sugar after ingestion of Mannan diet showed a maximum 10-minute increase of 10%, which is comfortably within the established safety limits for combined use with drug XYZ. Okay, so I don't do a lot of talking about what the lines are doing. What I'm talking about is what happened in the patient's body and reminding you the context of our story. We are not doing this to look at blood sugar and enjoy blood sugar changes. We are doing this to determine which of these is safe to use in patients who are taking this other drug. And so as we discuss these results, as we look at them, I want to discuss them in the context of that important story to make sure the audience keeps that in mind as we look at the data. This is, I think, one of the biggest difficulties when watching a presentation from a different field from mine. The people in the audience from the same field maybe can follow along the data, but I often have trouble knowing, what does this mean? How am I supposed to use this to answer I don't know the connection to the key question. I don't know how to feel about this graph. And so we need to make sure we discuss it in a way the audience keeps in mind the key story. You might think that's a bit long for me. So I made a shorter version as well. We can do the same thing in a shorter version. So here's version 2. The change in blood sugar level after ingestion of Mannin diet, represented by the pink line, is just what we are looking for: smooth and conservative, and at no point exceeding the safety limit of 15%. Sorry, I can't see the text because all this extra windows are open. At no point exceeding the safety limit of 15% increase or decrease in a 10-minute period. On the other hand, Slim and Slim, shown in blue, has a dangerous fluctuation of blood sugar, including rapid increases up to 20% in 10 minutes. Based on this, we can only recommend Mannin diet for patients taking drug XYZ. Okay, it's the same story, less details, but the message is still very clear, and it's still focused on the context of the story. Okay. Before we continue today, let's take a little break. Keep covering everything with these extra windows. It's 11:24, so let's say we start again at 11:30. So I'll put that into the chat. We're going to resume at 11:30, so let's take a short break. Two of you who are brave, if you'll turn your cameras, thank you very much, that's great. So I can see I'm talking to someone. Okay, what you see on the screen now is a pre-lecture practice that I gave you in the pre-lecture handouts. Hopefully, you took the time to read this and think about what do you believe is the main message for this passage. So after the first half of today's lecture, perhaps you can already predict what I'm going to tell you is the answer for this practice. And it's a little bit unkind practice to make you do this, but the answer for this is there is no way to know what is the message for this slide. So in case anybody didn't do the practice, let's read through this passage and think again about what do you believe is the key message, what the presenter wants the audience to remember from this slide. なぜか三人称になっちゃってる。This is a different version than the original, but that's not important. I think in the example I gave you, it was written as we did. In this example on the screen, it's Princeton researchers did. It's third person. Okay. Princeton researchers applied a strong magnetic field to the surface of a very pure bismuth crystal, perpendicular to the direction of electron flow. At very low temperatures, the relatively few electrons in bismuth were forced into orbits around any defects on the surface. This was the first time ever to directly image electron orbits in a high magnetic field. Despite their expectations that the crystal's lattice structure would result in 3 differently shaped orbits, the images revealed that all the elliptical orbits collectively aligned in the same direction. This technique is a promising tool to study these strange quantum fluids. Okay, so as I said, this is another example, sort of like the penicillin one, where there is simply no focus. So it's very interesting, but what happens as we listen to this is our brains begin to get overwhelmed, right? もう新しい情報ばっかりっていうか、脳の中がちょっと圧倒されてしまう。オーバーロードしてしまうんですよね。So we just keep getting new information, each new sentence. And we don't know, we cannot feel any natural focus. So whatever you chose as the main message for this will depend on you as the audience. It will depend on your own interest or background or where you tamatama happened to focus your attention in this passage. So I cannot tell you what is the intended message or the intended important idea in this passage. It's impossible to tell. So we need the presenter to tell us that. So different possible topic sentences for this could be, and any of these would be good, depending on what the real message is. Okay. A new imaging technique developed at Princeton has already provided some big results by confirming for the first time some old predictions. This is one of those partial conclusions. I don't give you the answers, but I give you a very clear indication of what kind of data, what kind of answers to look for or to listen for. Another one that could be good. Princeton researchers have developed a promising imaging technique to study the behavior of electrons in a quantum fluid state. Okay. For the first time, electron orbits in a high magnetic field have been imaged. That's possible. Thanks to a new imaging technique, Princeton researchers have confirmed a predicted behavior of electrons in a high magnetic field, but with unexpected characteristics. An unusual collective behavior in electrons in a quantum state was confirmed by a new imaging technique at Princeton. Or one more possibility. A team at Princeton has directly observed behaviors of electrons under high magnetic fields and demonstrated that the electrons in this state interact in a way that their orbits spontaneously align with each other. Okay. And the last one is a direct conclusion, but a lot of these are providing really clear listening goals, right? So these are all like good examples of topic sentences. But what is the real one? I don't know, because the original version does not provide us one. So that's why it is so important for you to make sure you know what is your message, what is your main character, and guide the audience with a topic sentence and by emphasizing the main character. Okay, moving on to, well, we're all in the same theme, but another important concept for starting your explanations is where to begin. So we need to create context to help the audience connect what they are hearing, the new information, with some knowledge they already have, something that they already understand, in order to help them comprehend and process that information. So where do we start? It kind of depends. So a lot of times, especially in biology, presenters will show like a tissue stain. They'll show like a slice of tissue and with lots of cells in it, and start talking about one part of that image. Say, as you can see, da, 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 da. Very often I feel I have no idea where I'm supposed to look. In this tissue, the presenter looks at this tissue and these cells every day. To them, it's like looking at a clock on the wall and knowing how to read it. But for me, I don't know. I see lots of colors and shapes. but I cannot see it the same way that they do. So just telling me, look here, I don't know what I am looking at. I don't know how it connects to anything that I have knowledge about. So it's very difficult for me to know how to look at it if we don't guide us there first. So it's beginning immediately with small, specific details can be very challenging for an audience from a different field. So what we want to do is we need to pick a starting point, somewhere that we know everyone in today's audience will be able to comprehend, and use that as a context to guide us to focus on that small part and understand how it fits in the bigger picture. So I'll give you an example here. Let's say that I am a presenter talking about where my special organism lives. So I'm going to show you today where I found, where I observed this very special organism that lives in a very specific ecosystem in Japan. So I think you know that Japan is located here in East Asia, and it's a group of islands. It has many islands, but right in the middle, around here, is a small group of islands called the Izu Islands. Going north to south, right around the middle here, you can see a very tiny island right here, and that is called Shikinejima. Shikinejima is known for these long, deep bays. And because of that structure, there are no waves inside, and the water is extremely clear. It's very good for snorkeling, but this is the only place that we can observe this organism in nature because of the clarity of the water. Okay, so that might be a way to introduce the location to an unfamiliar audience. Maybe if I am doing this in America or Europe or something. But you probably felt a little bit strange in today's audience, beginning with this here, showing you where Japan is. Now, obviously, everybody here is in Japan, so we know where Japan is. And for example, when I give this presentation at NIG, NIG is located in Mishima. Actually, Mishima is right here on this map. So people in Mishima, they already know about these islands. They have probably been there. So this would probably be the best place to start for an audience at Mishima, or where I live, because actually I can see this island from my house. Maybe for Tsukuba, I might start here. So I have to think about what my audience knows, what is their background, what is their familiarity, and start at the common point that I know everyone can be comfortable with, and go from there. So it's not so much big to small. For images, it's usually big to small. But the concept is really familiarity. What is the audience familiar with? So let me give you another example. And this isn't searching for the location, going big to small. This is understanding the role of a specific item or concept in a larger system. This is also something that's very challenging for people from a different field. You can describe, for example, a cellular mechanism, but people who aren't familiar might not know what is the purpose of that. How does that mechanism affect a larger biological function that I know about? If we can't imagine that, often it's very difficult to follow and appreciate a story that takes place on a very tiny level. So again, we want to try to introduce the role or the describe a piece of a system by choosing a reference point you know your audience can be familiar with. So I tried making some different versions to explain the role of this piece here. This is a gear, right? Hands of a gear. and I'm going to explain its role in the system. But I imagined 3 different audiences, all with different interests and different backgrounds. So this is a really important idea to plan for the specific audience and choose a reference point will be appropriate for them. The first audience I kept in mind was mechanical engineers. So maybe they're not specifically fans about bicycles, but there are some ideas or concepts that this audience should all know and appreciate and be comfortable using to start. So I chose this here, the importance of maintaining proper tension in a pulley system. This is a very basic concept for an engineer. I was an engineer in the past, so actually this is my favorite version, but I think you all might choose a different. Okay, version 1. It is always important to maintain a proper amount of tension in the bicycle chain to prevent it from falling off the gears. Because the radii of the different gears on the rear wheel are different, tension is particularly important when changing gears. To maintain this tension, there is a spring action idler pulley attached to the mechanism on the rear wheel that changes the gears, called the rear derailleur. The idler pulley is the lower of the 2 pulleys suspended at the bottom of this mechanism. Okay. The next audience I imagined is an audience of cyclists, people who their hobby is riding road bikes. And these people, I think you know, they generally maintain their own bicycles. They repair their bicycles, and they probably know very well the names and places of every part. So I'm just going to immediately start with, this is called the tension pulley, because I think they know that, and go to explaining its role from there. So version 2. All of you are familiar with the bicycle's tension pulley. It's the pulley on the bottom part of the rear derailleur, or rear gear changing mechanism. The pulley at the top moves to guide the chain from gear to gear, but the tension pulley also serves an important function. Each time you shift to larger or smaller gears, the length of chain needed also gets larger or smaller. Since the chain itself cannot change its length, the spring action tension pulley moves forward or backward to compensate and maintain the chain firmly in place. And the third audience I considered is people who don't really know much about bicycles, but probably have ridden a bicycle. In Japan, I think at least most of us have ridden a bicycle or know something about them. So I'm going to start with a very basic concept of changing bicycle gears. So I think most people can at least relate to that. So here's version 3. You have probably noticed that when you change gears on some bicycles, at the center of the rear wheel, the chain will jump from one gear to another. The biggest gear is easiest to turn, but provides little speed. As you go faster, you change to smaller gears, but that means there will be leftover or extra chain length. Under the gears, you can see a mechanism where the chain makes a zigzag path as it passes through 2 pulleys. The bottom pulley is spring-loaded and adjusts forward and backward to accommodate the extra chain length and prevent it from falling off. Okay, so those are 3 examples to talk about the role of the same thing, but assuming different backgrounds and interest in my audience, I used a different reference point to open each one. So in the chat, I would like to do a mini survey here. Which of these 3 explanations was most comfortable for you? Just 1, 2, or 3. I'll start. I already told you. I like 1. Okay, interesting. When I do this at Seoul Kendai, Sogo Kenkyu Daigaku in Daigaku, especially at Idenken or in Hayama, it's also biology. Basically, nobody chooses 1. Almost everybody chooses 3, and a few people will choose 2. And when I ask, usually the people who choose 2 are in fact cyclists. Today there's a little bit more variety, and I think it's because this course has people from different backgrounds. So in a case like this, what should you do? So it really depends on who you need or want to understand your presentation. But generally, assuming you have an audience of peers and you want them to provide feedback, we should aim for the field that is furthest away, but that we still want to follow our story. Sometimes, you might have guests or some outliers in your members from completely different fields who are just kind of interested. I don't think we need to plan the presentation for those people. If they're, just an outlier, they probably come to your presentation not really expecting to follow along, and that's okay. So maybe for an audience like this, I would choose 3 because it's the safer one and probably everyone can understand it. But depending on where I'm talking to, if I'm talking to my former classmates from university at engineering, I'm probably going to use 1. And if it's a group of cyclists, I will use 2. Okay. So our next practice, this is a listening practice. And I really want you to keep that in mind, listening. So those of you with biology backgrounds, you might be able to easily identify what should be the main character in this passage. But today I want you to turn off that scientific part for the moment and just use your ears. Tell me what vocabulary word, what single word do your ears pick up on very strongly? その単語の音がなんとなく目立つ。Okay? So as we listen to this, just choose the word that you pick up on. All mammalian brains have similar characteristics. The one on the left is from a mouse, and the one on the right is a human brain. A large portion of these brains is occupied by the neocortex, which is the superficial part shown here in green. A magnified image of this part is shown at the top. There are various types of neurons with different shapes and functions. These neurons are not randomly intermingled, but rather segregated into horizontal layers. For example, we can find larger output neurons in the deep layers and smaller intracortical neurons in the upper layers. In this way, the neurons coordinate their functions and are believed to account for mammals' higher cognitive level. This assumption is supported by the fact that neurons are not arranged in layers in the homologous brain region of non-mammalian species. Okay. So in the chat, I would like you to tell me what vocabulary, what word really stuck out in your ears. What did you hear very strongly? It's very cold coffee. Don't be shy, just whatever you heard very strongly. The answers I received so far are neuron, 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 and neuron. It's unanimous. Okay, so everybody picked up on the same word, neuron. And there's a good reason for that. It's because in the text, this is indeed, it stands out. It's very strongly emphasized because it appears in the strong position of many sentences. So in the beginning, it doesn't appear at first, right? The speaker starts with this pattern of introducing minor details, right? They give a very vague topic sentence. This topic sentence actually is not good. It doesn't reveal the main message. It reveals a very general topic, but not the message that we're supposed to learn from this. So that's not very good. And then we talk about, this is a mouse. This is a human. This is the neocortex. It's green. This is a magnified image. This is just like a tour guide guiding tourists around the slide and telling you what you're looking at. This is this is that. This is the kind of minor details with too much emphasis at the beginning that the audience doesn't need. We need to know what we're looking for and how to find it. So when the word finally appears is here, looking in the magnified image. There are 何かがあります. So what's the important position there? 何があるでしょうか, right? What is there? And the answer is various types of neurons. Okay, so neuron is in the strong position here. Next sentence. These neurons are not randomly. So the sentence is describing neurons. They are the subject and they are the strongest position. Next sentence. For example, we can find. We is the subject, but this is one that's the subject is not the strongest position. We can find. What do we find? This is what our brain is automatically listening for. 何が見つかるの? And the answer is we can find larger output neurons and smaller neurons. So again, neuron is in the strongest position. Next sentence. In this way, the neurons coordinate their functions. So here the subject with the important verb is again the strongest position, and it is neurons. Next sentence. This assumption is supported by the fact that, okay, so this first part is an introduction portion of the sentence. Then after that is another sentence, right? This is a kankeisetsu, a relative clause. So that neurons are not arranged in layers. Again, the subject and the strongest position is neurons, and the rest is describing the subject. So we see that this is a very powerful tool, this grammar of strong position. All of you notice the word neuron because it appears in the strong position. But that's not the main focus of this passage. And I realize now that on the slide, I already showed you the answer. I didn't mean to do this. Well, it's too late now. It's right here. So I was going to then ask you to turn on your scientific brain and see if you can tell me what should be the main character of this passage. And the main character is layered structure. Okay, it's the layered structure of the neurons. Well, that's too bad. I wanted to have you think about that. But so let's think about how we can improve this passage. First of all, I would like you to ask you to create a topic sentence. Okay, in the class, year-round class, 最近はちょっとなんか難しくやってないんですけれども、全体的に書き直してもらってたんですよ。結構難しいです。But only if you're a biologist and familiar with brains. So I will not ask you to rewrite everything, but I just want, that's the main topic of today's lecture. So I would like you to think what would be a better topic sentence for this passage? And I think this might be easier, again, in the small groups where you can discuss and share your ideas, especially if some of you are not very familiar with brain or some of the words involved here. So I'll put you into small groups again, and you can talk together and share your ideas about what you think would be an appropriate topic sentence. Remember what we talked about today. The topic sentence tells the audience what kind of information they're going to get, what they are supposed to learn, what is the important key message of this slide. Okay, for people who are not participating in the small group, you can type your topic sentence and just send it in the chat. There's a lot of you. I probably will not have time to respond to all of your individual ones. People in the group, if one person in the group will act like a secretary and type the topic sentence that your group creates, copy it, and when you come back to the main session, please paste it into the chat for the main session. Okay? So 各グループの中で一人だけは、みんなで決めたトピックセンテンスを、書き出して、それをコピーしといて、メインセッションに戻ったときに、そのチャットに、貼り付けてください。Okay, so I'm going to send you into the same breakout. I'm going to check to make sure the people are all still here. Sometimes it changes. I'm going to shift around a little bit. Okay. So please decide together, discuss together, what will be a better topic sentence. Let's look at the text. Sorry, just a moment. Sorry, when using a dual monitor, it's very difficult to follow my cursor from one monitor to the other. Okay, sorry. There in the chat, you have the screenshot of this screen with the image and the original text. This will help you, I think, decide the key question. Okay, I'm sending into the groups now.
So
thank you. I can
Okay.
go text. We have to make topic sentence. I just think we have to use word layered structure. Neurons are hmm? So if you don't mind, I write a topic sentence in chat area.
Okay.
So can I hear your opinion? Thought?
Hmm.
すみません、私本当に理系の知識がなくて全然わからないんですけど、
うん。
あれですよね。様々、ニューロンにバリアスタイプがあって、それがレイヤードストラクチャーであるっていうことで大丈夫ですか？それをさらにトピックセンテンスにしなきゃいけないんですよね。
うん。すみません。ちょっとさっき、ちょっと出てて聞いてなかったんですけど。
わかります？なんかトピックセンテンスをこの文章から作らなくちゃいけなくて。
そうですよね。はい。
はい。カーンさんがくれました。Human brain is supported. ですね。Layered structure of human brain is supported by neuron がミスター・カーンの意見です。
うんうんうんうん。
大体まとまってます。他に何か加えた方がいい情報ってありますか？
もう一回トピックセンテンス教えてもらっていいですか？
送りましょうか。ちょっと待って。
はい。
ですね。カーンさんが送ってくれたのですね。これになります。これできない。
Oops, news is different shapes from just means
これが今あるトピックセンテンスです。他に Do we need to add some other information to this sentence or not? A large portion.
なんだSo that means not only the human, but also human elephant. So all the mammals.
Okay. So you think we have to collect humans to mammalian?
Mammals, yeah.
Yeah, thank you.
Right?
I understand. Thank you.
Supported by new. Supported. Support. Assumptions. Supported. The meaning of supported is the same as a constructed. It is.
Okay. So we have to use other words. Not. Sorry, I just understand. Neurons are placed in layer structure, so its function properly is it meaningful. Like I just understand that. So what the appropriate word for supported?
Okay, support. This type of news. It is difficult to make
Yes.
a topic sentence. So neuron supported by different types of neurons.
Okay.
So, but it is good for topic sentence. Sounds like summary, right?
Okay. Okay, I
don't-
understand.
So because in the text, so they said the arrangement of different neurons make layers, right?
So,
It is true. Sorry, I didn't.
Ah. Layered. Dr. Khan says layered structure of mammal's brain have same characteristic.
So that is a layer structure.
Hmm.
So and we should include layered structure in the topic sentence.
Yes,
It is
I think
right?
so.
Okay. So layer structure of mammal brains. So okay. So Edi-san knows.
Sorry, what topic sentence we have now? Layered structure of mammal's brain is supported by different types of neurons. Is okay.
あれ
Not supported.
Last one from the Dr. Khan.
Layered structure of mammal's brain have same characteristics. Is topic sentence now.
Okay. It's same characteristics. Okay. Same characteristics include. Yeah. Thank you.
Thank you. Structure of
Brain has same characteristics. Okay. So the same characteristics means a layered structure and
okay.
different
So
arrangement of neurons. Okay. So probably it's fine.
So
Time has gone.
thank you. So I write last topic sentence in chat section. Thank you.
Thank you. Thank you, Khan.
Recording in progress.
Okay, everybody has returned to the main session. So if just one person from each group will paste into the chat what you decided as your key question, please put it in there. Okay, group 2 has put the role and benefit of the layered structure. Now, it's not quite a topic sentence just yet, but this would work absolutely as the main character for this. So we showed here that the main character should be basically the layered structure, but just like the penicillin, it's a little bit vague. So a more specific main character would be the role and benefit of the layered structure. Of course, it's not a sentence just yet, though, right? To make it into a topic sentence, we need to share with the audience what about the role and benefit. What is the role and benefit into a sentence structure? So you could say, for example, on this slide, I'm going to talk about the role and benefit of layered structure. But of course, いきなり layered structure って言われて何の layered structure わからないので so to make it a little bit easier, I'm going to talk about the role and benefit of having a layered structure of neurons in mammals' brains. That could be a topic sentence. Okay. Group 4 says neurons are effectively arranged in layers in the homologous brain region of mammalian species. Well, when we used homologous in the passage, we meant we were comparing mammals with non-mammals. All mammals have a neocortex, and this is what we were talking. We don't need the word homologous here. You can say the neocortex or just that they are arranged in layers. Neurons are effectively arranged in layers in the neocortex of mammalian species or in the brains of mammalian species would be good. Group 1. The layered structure of mammals' brains have the same characteristics. Have the same characteristics. The layered structure of mammals' brains have the same characteristics. Slightly vague, but I think it's enough for the audience to have a good listening goal, to know what to listen for as they listen to this slide. This could work. Okay. And group 3, in all mammalian brains, neurons construct a layered structure. As a native English speaker, construct structure doesn't sound good because we like to avoid the same sounds repeating, but neurons are arranged in a layered structure. Might be a little bit more natural. And that is also a good topic sentence. Okay. So I'm going to show you the example that I rewrote for this. And of course, not just the topic sentence, but the rest of the passage as well. So I have rewritten it to avoid the listing of minor details at the beginning of the passage, to include important context, important ideas, and reduce the minor details to supporting items inside the sentence. And then the other sentences, I'm going to take neurons out of the strong position and put the layered structure into. So here is that rewritten version. The layered structure in the neocortex is a characteristic shared by all mammalian species, including the mouse on the left and the human on the right. When the neocortex, this large green superficial part of the brain, is magnified, we can clearly see horizontal layers made up of neurons with similar shapes and functions. For example, the deep layers consist of larger output neurons, whereas the upper layers consist of smaller intracortical neurons. This ordered, layered structure is the basis for coordination of the neuronal functions and believed to account for the higher cognitive level of mammals. In fact, in the homologous brain regions of non-mammalian species, this layered structure is not found. Okay. So I intentionally rewrote this to include all of the same information from the original version, including talking about this is a mouse, this is a human, the green part is the neocortex, and this is a magnified image. Remember, in the original version, all of these were their own individual sentences. There was too much emphasis on this minor information. But now I've included it as minor clauses that fall in very weak positions. Right, so in the topic sentence we see the layered structure in the neocortex is a characteristic shared by all mammalian species, including the mouse and the human. So we're going to look at specific strategies for emphasis and de-emphasis. Is that tomorrow or next week? Next week. But I think you can see how this feels very small. In the next sentence, when the neocortex, this large green superficial structure here, is magnified, we can see. Okay, so our attention immediately goes to what do we see when we look at those places? And the answer to that is horizontal layers. Okay, so this minor information is included, but it's not calling the audience's attention and distracting them away. The strongest position has the key main character in it. So I think that listening to this, the audience will have no problem clearly catching the message for this slide, and that is that all mammals have a special structure where our neurons are in layers. And we think this is very important for coordinating the communication between neurons, and maybe that gives us an advantage over other non-mammals that don't have the same layered structure. Okay. So finally, today we're just going to look at one shorter example. I'm going to show you a before example, and then an after example, and then one more practice rewrite for you to do individually. It's a lot easier than the neuron example. Okay. This example here is from the saltwater frog, Ensei Kaeru no presentation, yesterday's video about the saltwater frog. So you have already seen this before. So the original version goes like this. The following slide shows the results of the experiment. Okay, this is not a topic sentence. Again, we hear this very commonly, but this is not the message of this slide. The figures shown here represent the disparity ratio of the actual surface area to the theoretical surface area predicted from the diameter. I think as the audience listens to this sentence, 一体何を聞いてるかわからないんですよね。かなりわかりにくい。It's basically defining the parameters that we are looking at, but suddenly this definition is too technical and very difficult for the audience to understand. It doesn't connect it to the bigger picture. There is no context to the story around it. It's suddenly very specific. The figures on the left are from untreated oocytes used as the control. The figures on the right are from the oocytes injected with LOL mRNA, which we call super LOL oocytes. Okay. This is not terrible, but what are we saying about the experimental group? These are the oocytes injected with LOL mRNA. Depending on your audience, this might be very difficult to understand. This injection of LOL mRNA is the procedure, the experimental procedure that was done. It is not what happens as a result of that procedure or our hypothesis, what we believe will happen as a result. And so depending on the background of your audience, do they know what it means to inject mRNA? And why do we inject mRNA? What do we expect to see from that? Now, of course, it depends on the explanation of the slide before. Maybe you have prepared the audience well for this, or maybe you have not. But this might be difficult to follow for some audience. And then on to the results. As you can see, the control oocytes have a ratio of just over 1. This means the cell is almost a perfect sphere with almost no membrane. The values for the super LOL oocytes, on the other hand, have ratios ranging between 3 and 4. Okay, but what are we talking about? Mostly numbers. We did kind of say this means it's a perfect sphere with no membrane, but how do we connect that? It's not directly explained. And finally, this means that although the cells appear spherical, they have a large amount of excess membrane. So the very last words we hear, they have a large amount of excess membrane. This is our main idea. This is the main message. That this procedure was indeed successful. Sorry, my voice is going. So the whole goal of this experiment was to achieve more membrane. And so we get that answer at the very end. It's very difficult to catch. So a better version of this would be the following slide shows that LOL overexpression did indeed increase the surface area of the oocyte membranes. The control untreated oocytes, shown here, were found to have almost no extra membrane. Comparing their actual measured surface area with the surface area calculated assuming they are perfect spheres resulted in a ratio of about 1. In impressive contrast, the oocytes overexpressing LOL or super LOL oocytes have gained a large amount of membrane, shown by the dramatic increase of the area disparity ratio to between 3 and 4. This reveals a large amount of extra membrane despite their spherical appearance. Okay, so quite a few changes I made. I get a topic sentence. I tell you from the beginning, success. It did indeed. Indeed implies that this was 仮説の通りですね. Our hypothesis was correct, right? We use indeed for that. So it did increase the surface area. We start with the control oocytes only. We use animation to focus the audience's attention on this. And we say first, they were found to have no extra membrane, the answer first, and then I explain how to see in the data that there is no extra membrane by looking at the ratio of actual membrane and the assumption that there is no membrane. And since the ratio is 1, it's equal. They have no extra membrane. Then I finally show you the experimental group, and I describe it not as injection of mRNA, but the result of that injection, which is overexpression of the gene. This is a more important concept for the audience to be thinking about. The result we see is a result of the gene overexpression, not a result of directly injecting, right? And I say first, again, before we talk about the values, first I say they have gained a large amount of membrane shown by the dramatic increase of the area disparity ratio to between 3 and 4. Okay, so hopefully you can see that this is going to be easier for the audience to comprehend what the message is and how to see it from the data. Okay. So our final practice today, I'm going to ask you to try to rewrite this passage, but like I said, it's much simpler. Okay. So here is a little background, just for those of you who don't know about transposons. So this researcher is looking at transposons in the genome of a plant. So transposons are basically short pieces of DNA that can jump around the genome. Usually these are bad, right? You have DNA bits randomly jumping around the genome. It can create lots of problems. So usually the organism has a way to protect against transposons. But so what a transposon is, it basically can move 2 ways. One, DNA portion can be copied and pasted into a new location via RNA. So whatever the original DNA bit is here, we might find the same bit appearing suddenly, pasted in different places. Or cut and paste, which means the DNA itself is actually cut out. It jumps from this location into other locations around the gene. So the presenter here, she knows the original position of the bits of DNA that are going to move. QTE1 is located here, which is the chromosome number 3 on this position. And QTE2, it's on chromosome 1 at this position. This is the original position where these bits of DNA start from. So she wants to know, where do the copies of these go? What's their target? Where in the plant's genome do these bits end up going to? And is there any pattern to the target? Specifically, do they prefer to go to these areas around the black circles here are called the centrosomes, the center of the chromosomes. Does it prefer this area, or does it prefer the arm regions? She wants to know this. So here is the original version. 実際に生徒の書いたバージョンですね。 My student wrote this original version. I investigated the integration sites of transposon QTE in this plant. まずこれは topic sentence ではない。This is not a topic sentence. Okay, this does not tell us the message or what we learned from this. It just tells us what she did. Okay, this figure shows the plant's chromosomes. These green and pink bars indicate the original sites where QTE1 and QTE2 were integrated respectively. The green arrowheads indicate the sites where the copies of the original QTE1 were integrated after activation. And the pink arrowheads indicate the integration sites for QTE2. As you can see, the transposons can be integrated in the pericentric chromatin regions and also in the arm regions. From this result, I could not find any preference for specific integration sites. Okay. So I will leave this on the screen. As a group, this is maybe a bit difficult, but individually, let's take about 15 minutes, I think, to rewrite this passage. Think about what is the main character. Make sure that that is identified in the topic sentence, that we get the main message of this slide in the topic sentence. And then as you describe the results, focus on the story, what we are looking for. Focus on the main character, not on green and pink bars and green and pink arrowheads. In this original version, these are very strong positions, but this is not a story about bars and arrowheads. This is a story about something else. If this is really difficult for you, just focus on rewriting the topic sentence. That will be enough. Okay. I will stay here if you have any questions. Thinking about everything we have talked today, try to write a better version of this slide's explanation. My students in the regular course also find this difficult to do, but it's very important, I think, to think about how to organize our grammar and our sentences in a way to position the important words and important topics in strong places, and to help maintain muishiki, subconsciously, the correct context, so that the audience can remember what is our story and how does this fit into our story. Basically, you can use very simple techniques of deleting and of rearranging to fix this passage.
What finish?
Just a few minutes. 5-10 minutes.
You are also on the workshop?
What?
You are also in the workshop, online workshop?
No.
We are late because we are in the workshop.
He workshop maybe his own way. He want to attend. Maybe.
Function.
No.
What about you?
I just like that.
We are
Don't worry,
on
don't worry.
It might still be a little early, but if you have finished, you can paste your rewritten version into the chat. If you're shy, you can make it a direct message to me so that other classmates can't see it. That's your decision. If you want to do it to Zane, to everybody, or just to me. Okay, there are about 3 minutes remaining. Just 3 more minutes. Okay, both Shindo-san and Kuroki-san wrote for the end, there is or there are any. I think you mean there are not any. ないってことですよね. There's no preference. There is not any preference or there is not any specific characteristics. Yeah, has no preference is correct, right? Okay, let's finish up in 1 more minute. Shindo-san, your topic sentence. Oh, that's okay. Sorry, it looked like 2 sentences. It's a comma, I guess, right? I wondered if there are any features on the Qt integration site. So I tried to find out by comparing the QTE1 and QTE2 insertion sites. Okay, so this is basically you're going for a partial conclusion, right? But maybe be a little bit more specific about what you mean by feature. And then I think it could be okay as a partial. For example, sorry, I can't read it. I wondered if there is any specific characteristic to the preferred integration sites of QTE. So to find out, I compared QTE 1 and 2 integration sites. That way you don't give us the answer, but you give us a clear indication of what kind of data we're going to get, and we have a good listening goal for how to get the answer. Okay, mostly I see lots of different ways of saying the same thing, and they all work. Okay, which is very nice to see. You're all choosing your own way to word it, and that's great. But basically, the first sentence, though, Shindo-san gave an example of how we could make a partial conclusion to give a hint about it. But most of you go for the full answer, which I think is probably appropriate here. Just remember that only saying what you investigated is not really enough. We need to have an indication of what did you find from that investigation in the topic sentence. If you reveal 100% of the details of your finding or not, that's your decision. Again, I would say for your principal experiment, the really key experiment, maybe you want to keep it slightly partial. But for any other, I think there's no reason to be secret about it. Just say the answer first. There's no preference, or they can integrate anywhere. So some of you said things like the integration sites were identified to be anywhere without a specific preference, or the transposon jump randomly on the whole chromosomal regions. Okay. Lots of different expressions for this. It's very nice. Most of you removed the specific sentences for "this is a plant" image. Okay. That kind of sentence is really unnecessary. So you can say things like the transposon can integrate anywhere on any chromosomes shown in the plant's genome here. Okay, something like that. So here is an example rewrite. This is maybe not my preferred version, but I wanted to keep it as close to the original as possible, so you can still recognize easily what I changed from the original version. So the conclusion first. I found that transposon QTE can integrate almost anywhere in the genome of this plant. The original integration sites of QTE 1 and 2 are shown by the green and pink bars, respectively, shown here in the plant's genome. After activation, they were copied and integrated at numerous sites on all chromosomes, as shown by the widely distributed green and pink arrowheads. As you can see, QTE could integrate in both the paracentric chromatin regions and in the arm regions. In other words, they showed no preference for any specific integration site. In this case, I used animation to make sure that the audience first can identify the original sites of the integration, and then when they know what they are looking for, I showed the data using animation so that they can react to it then. Okay, there are many, many different ways we could rewrite the same thing. I cannot tell you the way to say anything. Of course, speech, it's your style, it's your story. But remember that you're giving your story to the audience, hoping the audience will use this information to deeply comprehend and critique your research methods and your research results and interpretations. So we need to give our story to create our story and present it in a way that the audience can follow along as easily as possible and always catch the main message of each slide. So just please remember not to resist giving the answer first. I know it might feel unnatural, but that's okay. For a scientific presentation, once again, I'll say it's not literature, it's not beautiful, it's made for comprehension. We want to be impressed by the beauty of your experimental strategy, of your results, and your interpretations. Okay, so that concludes today's portion. And before I ask if there's any questions or comments, I will send in the chat now the PDF file for the slides for today. Sending now. Okay, so in the chat now, you should be able to access the PDF file for the slides of today with all the examples and the texts included for what we talked about in today. Tomorrow, we are going to talk about something I have mentioned many times already. I have talked about the importance of creating a logical connection between items in your presentation so that the audience is not surprised or confused when we see a new topic. It should always feel natural and expected. So tomorrow we are going to talk about how to logically connect the elements in your story and create this smooth flow to connect all of the ideas. Okay. Are there any questions or comments about today's topic?
すいません。口頭でも大丈夫ですか？
はい、大丈夫です。
途中そのニューロンの話の時にその文法的にだから目的語みたいなところとかあと there are とかああいうところが強調されるみたいなこと